Okay, so we have here the biochemical reaction. So biochemical reaction is one of the tests that would help us then to identify the bacteria that's been isolated from our clinical specimen. So biochemical reaction is just a chemical reaction, just like your chemistry test. So it's a chemical reaction between the products produced here by your bacteria. The products here could be in the form of the enzymes or even the end products of the different metabolic pathways that try to react here with a specific substrate found in our culture media to produce here reaction. Reaction here is pwede siyang colored reaction. Pag color reaction, mean to say change in the color of your culture media or even the appearance of the different color. Like your H2s production in the case of your production of your hydrogen sulfide with the blackening of their colony. Okay, so the result here could eventually be directly determined or by addition of your indicator. So indicator helps then to visualize the change in the reaction with your chemical reaction because of your bacterial products. So we'll be discussing here the basic principle and the different culture media utilized for each of the biochemical reactions and even the end product. Okay, let's just simplify the discussion for this. Okay, the first test here, we have the test for the indole. So indole here is a test for identification of the enzyme tryptophanase. So basic reaction for that, we have your tryptophan found in your trypton broth here. This is the culture media use, where you try to inoculate your organism in this culture media. Your trypton broth here contains your tryptophan, your amino acid. If your bacteria contains the enzyme tryptophanase, it will convert your tryptophan to your indole, and upon addition of your indicator in the form of your COVAX reagent, it form here the red color. Red is positive result, and yellow is the original color of the trypton broth, and therefore it's considered here to be negative result. Your COVAX reagent is your para-dimethyl aminobenzaldehyde. Okay, the next one, we have your MRVP or your metal red Voges-Proskauer. Okay, first we have here the metal red. Metal red here is a biochemical reaction that try to detect the products of your bacteria which try to convert your pyruvic acid to your mixed acid. Okay, the mixed acid here includes your formic acid, acetic acid, we have also here the succinic acid, and we have your lactic acid. So basic reaction, your bacteria try to utilize glucose as its carbohydrate source, which converted one to the pyruvic acid. But for some bacteria, they further convert the pyruvic acid here to your mixed acid. And that's what you are detecting in the metal red na test. Okay, the indicator for this one is your metal red. The positive result is red in color. Positive result, we have your Escherichia. Negative result, Klebsiella, Enterobacter, and we have also here the Serratia. Okay, the next related test for your MR, we have the VP or your Voges-Proskauer. Okay, the Voges-Proskauer is a test here that would detect stability of your bacteria to catabolize your glucose, converting that one to your pyruvic acid, and further convert that one to your acetoin, and oxidize to your diacetyl. Then you add your 40% KOH plus your indicator in the form of your alpha-naphthol to produce here your butanediol. Okay, so this is just, again, bacteria. It's try to identify bacteria which try to have another pathway for the utilization of the pyruvic acid. Let's try to convert that one instead to your mixed acid. Try to convert that one to your acetoin, your intermediate alcohol. Okay, this give a positive result here is still, okay, so indicator natin, so alpha-naphthol, positive result is red. So this includes here your Klebsiella, Enterobacter, Serratia. Negative is yellow. Again, ang rule natin for the MRVP, kung positive siya sa metal red, negative siya sa Voges-Proskauer. Kung positive siya sa Voges-Proskauer, negative siya sa metal red. So in the case of your Klebsiella, Enterobacter, Serratia, since they are positive here In the Vox-Boscawer, expect them to be negative in your metal red. Escherichia here is positive in your metal red, so expect to be negative with your Vox-Boscawer. Another test, we have here the citrate utilization. Some bacteria try to utilize citrate as their carbon source. Okay, Simon citrate agar is being utilized here as our culture media. Just need to inoculate your organism on that. Okay, indicator used here, we have your BTB or your brown thymal blue. Positive result would eventually be blue and negative result will be green for this. Okay, then we have also here the H2s, okay, or hydrogen sulfide production with the SIM. SIM stands for the sulfide. Okay, so that would detect here the three in the test. We have here the H2s production with the sulfide. We have also the I for the indole test, and for the M for the motility. Okay, so the indicator used here, we have your ferrous sulfate and sodium thiosulfate. So H2s production is identified here by the blackening of the colony. Okay, for the indole test, is what we have discussed kanina. So indole test will give a positive result with red in color. For the motility, so motility is identified here by the spreading of the colony from the area where you try to inoculate your organisms. Parang lumipat for example, kung dito ka inoculate, tapos parang pumunta doon ang mga colony niya, so mean to say that one is a motile. You try to spread to other area from the area where you try to inoculate. Okay, so this test will give you a 3 na positive result here. Test for the H2S, test for the indole, and we have also test for the motility. Okay, the next test, we have here your urea hydrolysis test. Test here would identify the enzyme urease. So if your bacteria contains the enzyme urease, okay, we have here your substrate urea. So your culture media for that is your Christensen's urea. Christensen's. Okay, Christensen's urea broth or agar. So it contains urea. If your bacteria contains the enzyme urease, it try to hydrolyze your urea to your carbon dioxide and ammonia. Indicator for that, we have your phenol red, positive results red, negative is yellow. Now we go to your carbohydrate fermentation. So this would identifies the ability of your organism here to catabolize or to utilize the different carbohydrates. So you could have here your lactose or you could also have here the sucrose. So for this one, you could have your lactose broth or your sucrose broth, inoculate your organism on that, then add your indicator in the form of your phenol red. Positive results yellow and negative result will be red in color. Okay, then we have also here the glucose fermentation. So again, this one would identify if your bacteria able to catabolize or utilize the glucose as its carbon source. Okay, then we have that glucose. So we have your glucose broth here. Okay, then you add your phenol red. Positive is yellow and negative is red. You can also add your Durham tube. Durham tube here, parang maliit na tube. Lagay mo dito sa loob. Okay, so that will be going to catch your air or your gas. That would help you identify if your bacteria would capable of the gas formation. The gas formation would be identified here as having the bubbles. So pag may mga space dito sa loob ng Durham tube or may bubble formation or space, then that's your bacteria capable of the gas formation. And then we have here your blood agar plate with the novobiocin. Novobiocin is antibiotic. So this will help you identify if your bacteria is resistant or susceptible with your novobiocin. That could be identified here by the zone of inhibition around your antibiotic disc. So if this one is your novobiocin, so try to stick here with the bacteria. Okay, so susceptible here is characterized by the presence of the zone of inhibition. So pag may space surrounding your antibiotic disc, so that's your susceptible. Pag wala siyang zone of inhibition, around your antibiotic disc, meaning to say your bacteria is resistant. Okay, then we have your MSA, mannitol salt agar. So, mannitol salt agar contains 7.5% sodium chloride. So, if try to trick your organism on that, so information can be derived out of this culture media. Number one, if your organism here able to grow in the MSA, so mean to say it able to tolerate here your 7.5% sodium chloride. Pag hindi siya nag-grow, meaning to say that one is being inhibited by your 7.5% sodium chloride. Remember, nag-grow lang dito ay Staphylococcus aureus. Another one would be, okay, dito also allows you to know here if your bacteria is capable of fermenting your mannitol or not. Okay, if your bacteria or your culture media contains here a yellow na colony, so that's mannitol fermenter. But if you have your pink na colony, your mannitol itself is pink. Okay, so if you have your pink na colony here, so mean to say that one is not a mannitol fermenter. Okay, another biochemical reaction, we have here the test for your coagulase. So coagulase is the enzyme that try to convert your fibrinogen to your fibrin clot. Okay, so there are two forms or two types of coagulase. One is your cell-bound or your cell-bound or clumping factor that could be detected here through your slide method. On the other hand, your extracellular or your free coagulase is detected here through your tube method. So your substrate for that would provide your fibrinogen, we have your rapid plasma. So since this one contains here the fibrinogen, if your bacteria contains the enzyme coagulase, the fibrinogen here will be converted to your fibrin clot, and therefore you're going to observe here for the clumping or coagulation. And that's considered here to be positive result. Okay, next we have here the biochemical reaction among your gram-negative bacteria. So kindly refer to a separate PowerPoint for this. The first one we have here for the carbohydrate utilization. Okay, for the carbohydrate utilization, we have here your lactose fermentation. Okay, so for the lactose fermenter, require here two enzymes. One is your beta permase. So the beta permase would allow here the permeation or the entry of the lactose inside your bacterial cell. And once inside the bacterial cell requires another enzyme, beta-galactosidase, which try to catabolize your lactose to your glucose and your galactose. So positive result is yellow, negative result here is red in color. Okay, lactose fermenter contains here two enzymes. So meron siyang beta permase and beta galactosidase. Your delayed lactose fermenter or late lactose fermenter only contains enzyme na beta galactosidase. Wala siyang permase. Kaya medyo nahirapan magkaroon ng reaction kasi wala siyang permase. So nahirapan pumasok ang lactose sa cell natin. In the case of your non-lactose fermenter, so wala talaga siyang enzyme. Wala siyang beta permase, wala lang siyang beta galactosidase. Okay, another biochemical reaction here with the use of this is your oxidation fermentation test. So this oxidation fermentation test here would be applied among our gram-negative na mga Enterobacteriaceae and other gram-negative rods or bacilli. Okay, your oxidation fermentation test here would have your two tubes. So this is your U live zone OF medium. So dalawang tube ito, one is left open. So bukas lang siya. The other one, lalagyan mo dito ng, okay, so if that's one, okay, the other one is being overlaid by your mineral oil. Okay, this one contains here your glucose. Okay, so that's 1% na glucose concentration and 0.2% na peptone. Okay, so this would identify here if your bacteria capable of oxidizing the glucose ba or fermenting your glucose. Okay, so original color of your tube here is green. positive result here would be your yellow. Okay, so your bacteria is a glucose oxidizer. Kung dito magpa-positive lang sa open at true, negative dito sa ating the one overlaid with your mineral oil. So, mean to say, pag oxidizer siya, dito ay yellow, ito ay green. Pag fermenter ang bacteria natin, nagpa-positive lang dito sa, okay, the one that's being overlaid with your mineral oil, negative dito. Mean to say, this is green, yellow dito. Okay, pag inert, on the other hand, so hindi siya oxidizer, hindi siya fermenter. So, both of it true would have your green na color. Okay, the next one, we have your TSIA or your triple sugar ion agar. So, this would have your triple because, again, this culture media contains here 3 sugars. And this is a special slant. Remember, your special slant would have here the butt area, and we have also the slant area. Okay, the butt area here contains the glucose at concentration of 0.1%, and the slant area contains here the lactose and the sucrose. That's why it gives here 3 na sugar. But the lactose sucrose in the slant area, that okay, will have a concentration of 1%. Indicator used here, we have your phenol red, and for the H2s production is being detected here by addition of your ferrous sulfate plus your sodium thiosulfate. Positive result is yellow. Pag yellow siya, that's acid or A. Okay, negative result is red. Original color nito kasi red. Okay, that's alkaline or K. Okay, the gas formation, on the other hand, is being detected by your crack in your culture media. Magka-crack siya, so that's gas. You could identify. So what are the results would be identified here with our TSIA? First one, we have A over A. So A, dito, that represent here the result of your slant area. Pag A ulit, that's your acid. Mean to say acid is positive. So pag acid siya, mean to say it try to ferment lactose and sucrose. Sa baba niya naman A ulit. So yellow ulit sa baba, yellow, yellow. And A here mean to say the glucose, positive siya. So yellow is positive. Mean to say it also try to ferment the glucose. May H2s, mean to say there's a blackening of the colony, may black ka nakita. Pag may gas, okay, that would identify here with a crack in your culture in the test tube or in the, dito, may crack dito. How about this one, if this is your result? K over A, pag K siya, mean to say the slant area is alkaline, mean to say that's negative. Mean to say this one do not ferment the lactose and the sucrose. Pag A sa baba, mean to say this one is acid, this is positive, signifying here that it ferment your glucose. So glucose fermenter lang siya for this one and is not capable of fermenting your lactose and sucrose. And wala siyang H2s, wala din siyang gas formation. Okay, another modification here of your TSIA, we have also the KIA or the Krigler iron agar. So this one differentiated here from the TSIA because this one only made up of 2 na sugar lang. Still in the butt area, we have the glucose, pero dito sa slant is only the lactose. Wala siyang sucrose. Okay, the next one, we have here the Onp Gina Test or the ortho-nitrophenylgalactopyrinoside. So this would detect the enzyme beta-galactosidase. Also for the lactose, fermentation pa din siya. Okay, so the substrate is the ONPG. If your bacteria contains this enzyme beta-galactosidase, it will convert your ONPG to your galactose plus your orthonitrophenol. Positive results, yellow. Pag yellow siya, it signifies that one is a lactose fermenter. Pag colorless, that's negative, then mean to say that one is not a lactose fermenter. Okay, then we have your biochemical reaction. Try to detect here your glucose metabolic end products. So we have your metal red, Vox-Pascour. We have discussed this already. So, okay. Okay na to. Next, we have here your amino acid utilization. So this is for some bacteria wherein they do not metabolize your carbohydrates, but rather just depend on the amino acid as their carbon source. So therefore, they try to utilize here the amino acid. So you can test that one, okay, by this test. So this is your decarboxylase na test. Bakit decarboxylase? Remember here that your amino acid made up of the carboxyl saka amino group, di ba? If your bacteria try to catabolize amino acid, utilize amino acid, it could catabolize that one by removing the carboxyl group if meron siyang enzyme na decarboxylase. Okay, amino acid, try to remove the carboxyl group by the enzyme decarboxylase sa mga ito, converting that one to the amine group or the diamine. Okay, example for that, you could have here your lysine. So if your bacteria contain the enzyme lysine decarboxylase, your lysine, as it try to catabolize this one, and try to remove the carboxyl group of the lysine, and try to convert it one to the diamine or the amine group in the form of your cadaverine. Ornithine naman, pag ginagamit ng ornithine, try to catabolize that one with the enzyme ornithine decarboxylase that will be converted to your putrescine. And arginine dihydrolase naman, if try to utilize arginine, converting that one to your citrulline, and finally to your ornithine. Okay, the content of your, so tatlo ito na test tube. Okay, one is a test for the lysine, the other one is a test for the ornithine, and a test for your arginine. Okay, so each of this one, okay, na culture media dito ay it contains here 1% of your amino acid. If this one is your lysine, so 1% na lysine, ornithine, so 1% then, and your arginine is also 1%. Then we have also here the indicator used. We have here your brom crescent purple. Positive result is violet or purple in color. Negative result, we have here yellow na color. Okay, so we have here some of the result with your decarboxylase test. So we have here your Klebsiella pneumoniae, Oxytoca, Enterobacter aerogenes, Enterobacter cloacae, Proteus vulgaris, and we have Proteus mirabilis. So for the lysine decarboxylase, nag-positive lang dito ang dalawa nating Klebsiella pneumoniae saka Oxytoca plus your Enterobacter aerogenes. In the ornithine decarboxylase, na positive lang dalawa natin na Enterobacter o aerogenes sa cloacae plus your Proteus mirabilis. But in case of your arginine dihydrolase, ang positive lang dito is your Enterobacter cloacae. Okay, the next test, we have here the Amines Test. So kanina, decarboxylase, ngayon the Amines. So these are the bacteria we try to catabolize our amino acid by breaking down the amino group. Kanina carboxyl group, kaya deaminase ito. So, this one, try to utilize the phenylalanine as the amino acid. If that organism contains the enzyme phenylalanine deaminase, it try to remove the amino group, it will be converted to your phenylpyruvic acid. Indicator for that, para magkaroon ng reaction, ferric chloride. Positive results, green. Negative is yellow color. So positive result here includes the group or the family of your Proteus, includes your Proteus morganella, and we have also here the Providentia. Okay, now we go to the miscellaneous test. So we have your citrate, malonate, and acetate. So some bacteria try to utilize citrate, malonate, or even acetate as their carbon source. So we have discussed already your citrate utilization. So bromthymol blue, Simon citrate, bromthymol blue. Ang kanyang indicator, positive result is blue, negative result is green. Another one, we have here the enzyme denase. Sa bacteria would have this enzyme, we try to catabolize your DNA, converting that one to your short oligonucleotide. Then you add 0.1 hydrochloric acid, clear. Maging clear ang culture media natin. That's positive result. So, denase na enzyme could be found here with your Staph aureus, and we have your Serratia marcescens. Another one, we have your gelatin liquefaction. This would identify the enzyme gelatinase. So, ang ating culture media is your gelatin bot. Okay, so inoculate natin ang organism natin. If your organism contains this enzyme gelatinase, it will liquefy your gelatin. Remember that your gelatin is solid. So, pag may enzyme siya na gelatinase, magiging liquid ang ating gelatin. So, through liquefaction with the enzyme gelatinase could be identified here if you try to ref your gelatin bot. If you try to ref that one, so kung hindi siya mag-solidify, so it's a true liquefaction because of the enzyme gelatinase. However, pag solidify ulit siya upon refrigeration, meaning to say that one is not a true liquefaction. Okay, another test here, we have your indole. Okay, indole production, as what we have discussed already, would identify here the enzyme tryptophanase that would base on the ability of your bacteria to utilize tryptophan, converting that one to your indole. Positive result is your red in color upon addition of your COVAX reagent. Okay, another test, we have the nitrate reduction that would test ability of your bacteria to convert your nitrate with the enzyme nitrate reductase to your nitrite. Upon addition of your sulfonilic acid, it will give a pink na color reaction as positive and yellow is negative. So pag nag-yellow siya, we need to confirm by addition of your zinc dust. Upon addition of your zinc dust, pag nag-pink siya, mean to say that one is a true negative reaction. Nag-pink siya because this is your positive control. Pag nag-pink na siya after addition of zinc dust, mean to say that one is your true negative result. Nag-positive siya is because nag-add ka ng ating positive control. Pag nag-yellow pa din siya, even after addition of your zinc dust, which is your positive control, it will signify here that the bacteria present is capable of reducing your nitrate to your nitrite, meaning to say meron siyang nitrate reductase. However, pag na-convert na niya sa nitrite, it further convert to your nitrogen gas. So kahit anong gawin mo, magne-negative pa din siya even if you add this one because again, this is a bacteria which capable of converting it further to your nitrogen gas. And now we go to the different miscellaneous tests. So we have your urease test. We have discussed this already ulit. Diba? So that's your urea. Urea is carbon dioxide ammonia plus your phenol red. The red is a positive result. Another test, we have the LIA or your lysine iron agar. So this is the culture media wherein this is also a special slant. Okay, the slant area would identify the enzyme na lysine deaminase. And the butt area would identify the enzyme lysine decarboxylase. So it depends on the bacteria kung kaya niya i-deaminate ba, remove the amino acid, or remove the carboxyl group. So you could identify that one with your butt in a slant area. Okay, the lysine incorporated for this one is 1%. You have glucose also. Glucose is being incorporated also here, lysine at 1%. Indicator 4, we use here, we have your brome crystal purple. And we have also here for the H2s. So, H2s production, we are using ferric ammonium citrate plus your sodium thiosulfate. Okay, the slant, ang positive result natin that would identify your enzyme, lysine, deaminase, is your burgundy or plum or red na color. Negative is yellow. The butt area would identify the enzyme lysine decarboxylase. That would have here purple na color. Negative result for that is also okay, yellow na color. Okay, so like for example, if you have the test result here, so Y over P, Y is yellow, P is the purple. Ano ibig sabihin nun? Pag Y dito, mean to say yellow, mean to say wala siyang enzyme na lysine deaminase. Pag purple dito sa baba, sa butt area, mean to say meron siyang enzyme na lysine decarboxylase. H2S, that would be manifested here with the blackening of the colony, or you could also have gas formation. Kung may gas siya, then we have your crack in your culture media. Another test, we have your motility indole ornithine. Okay, so this is a 3-tube na test that would identify the test result for your motility. Motility would be identified here by the spreading of the colony from the area where it tried to inoculate your organisms. Indole here would give positive result upon addition of your COVAX reagent, red color. And ornithine, that's ornithine decarboxylase, so kaninang tatlo, ornithine, lysine, they would have here positive result for the purple. Another one, we have the SIMS. So in same here for sulfite, H2S, with the blackening of the colony indole for the purple color. The motility would be identified here by the spread of your colony from the inoculated area. Okay, another biochemical test, we have here the oxidase test. So this test would identify the enzyme cytochrome oxidase. The substrate is your hydrogen peroxide, converting that one to your carbon dioxide and water. So our indicators, we have tetramethyl phenylenediamine dihydrochloride. Positive result purple that usually occur in your Pseudomonas, Neisseria, Micrococcus. Colorless is the negative result that will be found here with your Enterobacteriaceae and your Staphylococcus. Okay, so thank you.